Hello everyone, welcome to our channel Biological Concepts. In today's video, we will discuss some important questions on bacterial stainings. Some brief notes regarding the questions and their related options are also provided here, which will be beneficial for you. So stay tuned in this video. Basically, staining and fixation is used to increase the visibility, accentuate specific morphological features and preserve them for future study. Now, let's discuss the first question. If only one stain is used to stain the specimen, then it is known as option 1 simple staining, option 2 differential staining, option 3 gram staining, option 4 is negative staining. And the correct answer is option 1 simple staining. Now, in simple staining, only one stain is used and all types of bacteria appear as the color of that stain when viewed under the microscope. So when this bacteria are stained by simple staining, then the bacteria appear as the color of that stain, particular stain, when viewed under the microscope. The stain used in a simple staining are crystal violet, safranin, methylene blue, etc. It is used to determine the bacterial species morphology, that is cell shape, whether it is cocci shaped or spherical in shaped, whether it is rod shaped, etc. And also determine the arrangement, that is if it is single form or in chain formation or in cluster formation, etc. But this simple staining do not give any additional information. Okay. It only determines the bacterial species, morphology and arrangement. Okay. When the simple staining is done by crystal violet, then the cell looks like violet in color. When it is stained by safranin, then it looks like pink. Okay. It will be pink in color. And when it is stained by methylene blue, then it will be in blue color. Okay. Next question. If more than one stain is used, then the staining procedure is known as option 1 simple staining, option 2 differential staining, option 3 counter staining and option 4 is negative staining. And the correct answer is option 2 differential staining. In differential staining, more than one stain or dye is used. More than one stain or dye is used to stain and differentiate different group of bacteria. This is in contrast to simple staining, where only one stain is used. Okay. In differential staining procedure, it takes the advantage of the variation in the chemical and physical properties of different groups of bacteria. The examples of differential stainings are gram staining, that differentiate between gram positive and gram negative bacteria. Endospore staining, which differentiate the vegetative cell from the endospore, and the acid fast staining, which differentiate between acid fast and non-acid fast bacteria. Next question. When other than the sample, that is the specimen, the remaining portion is stained, then this staining procedure is known as option 1 simple staining, option 2 differential staining, option 3 negative staining and option 4 is gram staining. And the correct answer is option 3 negative staining. So, in negative staining, the sample is not stained, rather the remaining portion is stained. It is performed for the observation of intact microbial structures without disturbing its cellular morphology. It is referred to as negative staining because the bacterial cells are not stained. Rather, the background is stained. Whereas in simple staining, differential staining, the bacterial cells are stained, not the background is stained. Okay. So, in negative staining, the bacterial cells are not stained, rather the background is stained. Okay. Now, what are the advantages of using this negative staining? These are: cells appear less distorted because no heat fixing is done. The heat fixing is done in case of simple staining, differential staining. But in negative staining, no heat fixing is done. That's why cells appear less distorted. Second is capsulated bacteria, which are difficult to stain, can be observed by this technique. So, by this negative staining, capsulated bacteria are stained. Next question. Acidic dyes binds to option 1 negatively charged protein, option 2 nucleic acid, option 3 both 1 and 2, option 4 none of the above. And the correct answer is option 4 none of the above. So acidic dyes doesn't bind to the negatively charged protein or the nucleic acid. Now these acidic dyes are negatively charged. That's why they are unable to bind to the negatively charged protein or nucleic acids. They only bind to the positively charged cell structure. Examples of acidic dyes are acid fuchsin, eosin, rose bengal. Whereas the basic dyes are positively charged. They bind to negatively charged molecule like nucleic acid, many proteins and surfaces of prokaryotic cells. The examples of basic dyes are safranin, methylene blue, crystal violet, basic fuchsin, malachite green, etc. Next question. Heat fixation preserves option 1 overall morphology, option 2 internal structures, option 3 both 1 and 2, option 4 none of the above. And the correct answer is option 1 overall morphology. So heat fixation preserves overall morphology. Now what is fixation? Fixation is the process by which internal and external structures of the cells and microorganisms are preserved and fixed in position. This fixation is two types. One is heat fixation and another one is chemical fixation. Now this heat fixation preserves overall morphology. It is used in biology labs to prepare bacterial smears for staining. Typically, a film of cells or smear is gently heated as the slide is passed through a flame. This heat fixation is used in gram staining, endospore staining, etc. Next one is chemical fixation. This chemical fixation used to protect the fine cellular substructure, the morphology of larger and more delicate microorganisms. Next question. Capsule staining is option 1 simple staining, option 2 negative staining, option 2 differential staining, option 3 negative staining, and option 4 is gram staining. And the correct answer is option 3 negative staining. Now we need to utilize negative staining technique to view the outline of the capsule. In negative staining, it uses the acidic dye, which are negative charged. The negative stains are repelled by the negatively charged bacterial cells, preventing the cells from taking up the stain. Therefore, in negative staining, the background is stained rather than the microorganisms. Okay. So, because of the negative charge of the stains, these are repelled by the negatively charged bacterial cells, so that they prevent the cells from taking up the stain. That's why in negative staining, the cells does not take the cells or microorganisms are not stained. Rather, the background is stained. Next question. Sudan black is used to stain option 1 protein, option 2 lipid, option 3 DNA, and option 4 is RNA. And the correct answer is option 2 lipid. Sudan black, also known as Sudan 3, selectively stains lipid because it is lipid soluble but will not dissolve in the liquid portion of the cells. So it is used as a stain of lipid. Next question. Endospore can be stained with option 1 crystal violet, option 2 malachite green, option 3 methylene blue, option 4 is basic fuchsin. And the correct answer is option 2 malachite green. This endospore staining where malachite green is used as staining agent is known as Schaeffer-Falter method. In this method, primary stain malachite green is used. As mordant, it is used. whereas as counter stain, safranin is used. By this Sheffer-Fulton method, the endospore appear as green in color, whereas the vegetative cells appear red in color. Next question. In gram staining, the gram-positive bacteria appeared as option 1 pink, option 2 green, option 3 violet, and option 4 is red. And the correct answer is option 3 violet. In gram staining procedure, the primary stain used is crystal violet, whereas as mordant, iodine is used. The counter stain safranin is used. The gram-positive cells take the primary stain and appear as crystal violet and appear as violet in color, whereas the gram-negative cells appear pink in color because it's on after decolorization it loses the CVI complex, that is the crystal violet iodine complex, and takes the counter stain color, that is safranin. That's why the gram-negative bacteria appear pink in color. This is the gram-positive bacteria after staining, gram staining, and this is gram-negative bacteria after gram staining. Next question. The Fulton procedure is used to stain option 1 DNA, option 2 cell membrane, option 3 cell wall, and option 4 is none of the above. And the correct answer is option 1 DNA. This procedure is discovered by Robert Fulton, and according to this Fulton, this procedure is named, that is Fulton procedure. It is used in histology to identify chromosome material or DNA in cell specimen. The stain is darkly stained. The staining compound, that is a sweep reagent, is covalently attached to its deoxyribose sugar. It depends on acid hydrolysis of DNA. Therefore, fixing agent using strong acid should be avoided. Next question. In gram staining, dash is act as mordant. Option 1 iodine, option 2 malachite green, option 3 safranin, and option 4 is heat. And the correct answer is option 1 iodine. So in gram staining, iodine act as mordant. Now what is mordant? A mordant is a chemical agent that keeps the dye in place. It holds the molecule of a stain onto the microorganisms. Anything that holds down a dye, be a metal ion or a halide ion, can be classified as mordant. In gram staining, the iodine complex by iodine form complex with the crystal violet in the bacterial cells and form CVI complex, that is the crystal violet iodine complex, and hold this CVI complex into the cell wall and appear as violet color after decolorization. Next question. The ratio of acid alcohol used in acid-fast staining is option 1 is 3% HCl and 95% alcohol, option 2 5% HCl and 95% ethanol, option 3 3% HCl and 97% ethanol, option 4 4% HCl and 96% ethanol. And the correct answer is option 1, that is 3% HCl, that is hydrochloric acid, and 95% ethanol. This acid alcohol act as decolorizer in acid-fast staining. Whereas in Endospore staining, water acts decolorizers, and in gram staining, either 95% ethanol or acetone act as decolorizing agent. Next question. In acid-fast staining, the acid-fast bacteria appeared as option 1 red in color, option 2 blue in color, option 3 green color, option 4 pink color. And the correct answer is option 1, that is red, or it may be appear as pink in color. The acid-fast bacteria example is mycobacteria. Acid-fast bacteria are known as acid-fast due to the presence of mycolic acid, whereas non-acid-fast bacteria appear as bluish purple after acid-fast staining. The order of staining in gram staining procedure is option 1 crystal violet, followed by safranin, followed by alcohol, followed by iodine. Option 2, crystal violet followed by alcohol followed by iodine followed by safranin. Option 3, crystal violet followed by iodine followed by alcohol followed by safranin. Option 4, crystal violet followed by alcohol followed by safranin followed by iodine. And the correct answer is option 3, that is crystal violet followed by iodine followed by alcohol. Last one is safranin. Crystal violet acts as primary stain, then iodine acts as mordant, alcohol as decolorizing agent, and safranin is counter stain. On acid gram staining, the gram positive bacteria appear as violet in color and gram negative bacteria appear pink in color. Next question, in acid first staining, acid firstness of the bacteria is due to the presence of option 1 mycolic acid, option 2 lipid, option 3 protein, option 4 is plasmid. And the correct answer is option 1 mycolic acid. The acid firstness of mycobacteria is due to the high mycolic acid content of their cell walls, which is responsible for the staining pattern of poor absorption followed by high retention. Some bacteria may also be partially acid first, such as Nocardia, whereas mycobacteria is totally acid first bacteria. Next question, the order of staining in acid first staining procedure is option 1 purple fuchsin followed by acid alcohol followed by methylene blue, option 2 methylene blue followed by acid alcohol followed by carbon fuchsin, option 3 malachite green followed by acid alcohol followed by safranin, option 4 crystal violet followed by acid alcohol followed by safranin. And the correct answer is option 1, that is carbon fuchsin followed by acid alcohol followed by methylene blue. This acid first procedure is known as gel and nail salt staining procedure. Here as carbon fuchsin used as primary stain, as decolorizer acid alcohol is used, 3% HCl and 95% alcohol. And as counter stain, either methylene blue or malachite green may be used. Next question, the order of staining in endospore staining procedure is option 1 malachite green, heat, water, safranin, option 2 safranin followed by water followed by heat followed by malachite green, option 3 malachite green, acid alcohol, safranin, option 4 acid crystal violet followed by acid alcohol followed by safranin. And the correct answer is option 1 malachite green followed by heat followed by water followed by safranin. This procedure is known as Schaefer-Fulton method. Here malachite green act as primary stain, heat act as mordant, water act as decolorizer, and as counter stain safranin is used. And the endospore appear as green in color upon endospore staining, and the vegetative cells appear as red on endospore staining. Thank you. If you like this video, please like, share and subscribe to this channel. Thank you.